Hello, everyone. Thank you for attending today's webinar. I am Sophia, the head of sales and marketing at Aussie Biosciences, and I am today with Cedric.
Hello, everyone.
From Aussie Biosciences also, and Marcelina from ExploRNA.
Hello, everyone.
So I'm going to launch the presentation. If you have any question, do not hesitate to ask them during the presentation, and we will try to answer them as best as possible at the end. And we also have a quick form for you to fill in a quick satisfaction survey at the end. So we would be really glad if you could stay until the end and answer this question. So I'll launch the presentation.
Aussie Biosciences is a French biotech company founded 20 years ago, specialized in molecular delivery systems present in France and the US. The French site is located in Marseille and comprises the headquarters and the production site, divided in 2 laboratories of over 500 square meters. It is responsible for research and development and production, mRNA and LNP platforms, marketing, and sales. The US site is installed in San Diego and deals with marketing, sales, and business development. Over the years, we have settled a worldwide distributors network. One of our objectives is to develop the spread of our reagents and expertise. This expertise is based on more than 20 years of experience and innovation. Several important milestones have marked the company's history, such as the commercialization of the first magnetofection reagents in 2004, the creation of OZB Inc. in 2013, and our recent acquisition of new facilities for the development of LNP and mRNA platforms. We are proud to cover a huge variety of fields, from bioproduction to viral applications, going through in vivo protein delivery or stem cell applications. All the products developed and sold, as well as our platforms, are under the ISO 9001 certification. These innovative reagents allow Aussie Biosciences to offer solutions in 3 main areas: cell and gene therapy, bioproduction, and custom platform productions. We're going to focus on our catalog mRNA and more precisely on our mRNA production platform. We provide a full mRNA custom service from sequence to final product. And during this webinar, we'll be covering in detail the cap analog ExploCap, ExploRNA's latest innovation in co-transcriptional 5' capping technology. This is an exciting addition to our platform that opens new possibilities in terms of mRNA translational performance. But first, let's have a look at our mRNA production. Our mRNA production platform is built around a tightly optimized and controlled 5-step workflow. Starting from gene synthesis and cloning, that is followed by plasmid preparation, linearization, and purification, a crucial step for high-quality subsequent RNA. The next step is in vitro transcription, or IVT, which is the enzymatic synthesis of the mRNA molecule itself. Capping can be done enzymatically or co-transcriptionally using ExploRNA's proprietary technology that we will describe later. Step 4 is purification and tangential flow filtration, or TFF, that removes reaction byproducts and concentrates, the final mRNA. The produced mRNA are finally qualified by an extensive quality control. The basis of mRNA is the DNA template. We use our own optimized expression vector in which the sequence of interest, or ORF, open reading frame, is subcloned. This plasmid is designed for high-efficiency mRNA production. The vector architecture is carefully structured. Moving from the 5' end, the T7 promoter sequence will position the T7 RNA polymerase for efficient transcription initiation. This promoter is followed by a 5' UTR sequence that has been optimized to maximize ribosome engagement and translation efficiency. Right after, we insert the ORF coding for the protein of interest that is followed with an optimized 3' UTR, playing a key role in mRNA stability and translational regulation. And finally, a poly A tail is encoded directly within the DNA template. Once the gene of interest is subcloned into this vector using the appropriate restriction sites, the plasmid undergoes linearization. Restriction enzyme cuts at the end of the poly A sequence to ensure that transcription terminates at the right position, producing an mRNA of defined length. The linearized plasmid is then purified to remove enzymes and buffer components before proceeding to in vitro transcription. IVT, or in vitro transcription, is the core enzymatic step that converts the DNA template into a functional mRNA molecule. It is a multipartite reaction that comprises the linearized plasmid template designed before, T7 RNA polymerase, a complete set of ribonucleoside triphosphates, or NTPs, cap analogs for co-transcriptional capping, and an optimized buffer system. The reaction proceeds through 3 classical phases. During initiation, the T7 polymerase recognizes and binds the promoter sequence, positioning itself to begin synthesis. Then, with the elongation step, the polymerase reads the DNA template strand in a 3' to 5 direction and sequentially adds complementary ribonucleotides, synthesizing the mRNA in a 5 to 3 direction. Finally, the termination step, when the polymerase reaches the end of the linearized template, the synthesis stops and the full-length mRNA molecule is released. The DNA template is then digested using DNA cell, and resulting RNA is purified to remove free nucleotides, fragment of DNA template, residual proteins, and reaction byproducts, giving us a clean mRNA product ready for downstream processing. The IVT is a classic process to create mRNA. So what makes OZ Biosciences different? We've optimized every single component of the IVT reaction. Starting with modified nucleotides, we offer 100% replacement with a range of clinically relevant analogs: N1-methyl pseudouridine, 5-methoxyuridine, 5-methyl CTP, or others for custom applications. We also have ameliorated the process to propose the incorporation of fluorescent NTPs for tracking applications, for example, and various chemically modified NTPs for RNA functionalization. Then we use an optimized T7 polymerase engineered to minimize double-stranded RNA, or dsRNA, formulation. dsRNA is a potent activator of innate immune sensors, and its presence in the final product can trigger unwanted inflammatory responses and reduce translation efficiency. Our enzyme also delivers high mRNA yields with no short abortive transcripts, which is essential for downstream purity. Next, our buffer is optimized and includes ribonuclease inhibitors to protect the mRNA from degradation during synthesis, as well as inorganic pyrophosphatase, which removes pyrophosphate, a byproduct that would otherwise inhibit polymerase activity. For the poly A tail, we encode the poly T stretch directly into the DNA template, ensuring a defined homogeneous tail length. Finally, for capping, we offer 2 approaches: enzymatic capping to generate cap0 or cap1 structures, as well as co-transcriptional capping using ExploCap analog, which allows efficient and scalable capping in a single reaction step. We will discuss more in detail about this cap analog in the following. All of this is combined with a robust purification process and tangential flow filtration to develop mRNA of the highest purity and consistency. First, let's have a look at our mRNA and saRNA that are ready to use and available in less than 48 hours. Our catalog mRNA span from reporter gene encoding fluorescent and luminescent proteins to genome edition mRNA, cytokines, or transcription factors. Our self-amplifying RNA comprises reporter genes and vaccine or antigenic transcriptions. Our catalog mRNA have been validated in numerous publications, such as this study from Jin Feldner in NPJ Vaccines in 2025 that had an objective to develop and optimize lipid nanoparticle formulations for a bivalent mRNA vaccine that the authors called Fluvid vaccine. This vaccine would target simultaneously two major respiratory pathogens, influenza A, H1N1, and SARS-CoV-2. The article used 7-week-old mice immunized via intramuscular injection as a model for human mRNA vaccine, and 3 different modified mRNA were compared. These modifications differentially modulate innate immune sensing and would induce different antibody responses, as shown on the graph, where anti-spike IgG were measured several days post-injection. Cytokine profiling was performed on blood samples collected at 3 hours post-prime on day 0 and 3 hours post-boost on day 14. The results show that mRNA alone or combined elicit a cytokine production in the same range than protein vaccine. Specific antibodies dosage confirmed cytokine observation. One of the most striking results was that both unmodified mRNA formulations conferred 100% protection against viral challenges, whether the antigens were delivered as separate monovalent constructs or combined in the bivalent Fluvid vaccine. More importantly, the combination of the 2 mRNAs into a single LNP formulation provided dual protection without reducing efficacy. This article definitively correlates the superior purity of the mRNA supplied by OZ Biosciences with the 100% protective efficacy. As the author stated, bioanalyzer profile indicate pure products of mRNA and similar levels of protein expression were detected from HEK293 cells transfected with these mRNA products, confirming that mRNA purity directly translates into biological performance. In support of our catalog offer, we can also provide high-quality custom mRNA for any gene of interest. And we have made our mRNA platform as flexible as possible. Every element of the mRNA can be tailored from the 5-cap to the poly-A tail. We can replace nucleotides with modified ones and also fluorescently label your mRNA. We can also adapt to your starting material. Option 1, you simply provide us with your gene sequence in FASTA format. We take it from there, gene synthesis, cloning into our optimized vector, and all the steps we've described before. Turnaround for this option is around 6 weeks. Option 2 is faster, about 2 weeks, and it's for customers who already have a DNA template ready to go. In that case, we just need you to meet 3 requirements. First, the plasmid must be linearized, or as a circular plasmid with a well-defined restriction site. Second, your construct should bear a T7 promoter and a poly-A tail. And if the poly-A tail is absent, that's something we can add as an additional service. Third, the DNA needs to be properly purified. We're looking for an A260-A280 ratio above 1.8 and an A260-A230 above 1.8 as well, using standard purification methods such as phenylchloroform extraction or column chromatography. In both cases, the minimum input is 20 micrograms of linearized DNA, and from that, you can typically expect approximately 1 milligram of purified mRNA in return. With more than 20 years of experience, we're confident in providing you with high-quality mRNA and saRNA. We ensure that all mRNA and saRNA meet our drastic criteria, including mRNA integrity with length, poly-A tail and capping validation, and purity and sterility, as well as functionality when possible. We also offer a superior grade quality control with the measurement of endotoxin and dsRNA amounts. We will now give the hand to Marcelina from ExploRNA to introduce their cap analog technology.
ExploRNA Therapeutics is a biotechnology company pioneering next-generation mRNA technologies for vaccines and therapeutics. Founded in 2019, the company combines deep scientific expertise in RNA chemistry, cap analog innovations, and in-house R&D capabilities to develop advanced solutions that enhance mRNA translation efficiency. We are located in the heart of Poland, with laboratories in Warsaw and Białystok, where our cap analogs are developed, manufactured, and tested. With our strong IP portfolio and a library of over 150 capping reagents, ExploRNA actively collaborates with partners across the US, Europe, and Asia to advance mRNA technology. After this short introduction, I'd like to present our core technology. So let's start with the basics. Probably all of you know that every mRNA molecule encoding a specific protein exhibits a characteristic and very well-defined structural architecture. A key distinguishing feature is the presence of specialized structures at its 5' and 3' termini. Today, for obvious reasons, I will focus on the 5' end of mRNA, so-called the cap structure. It consists of a chemically modified 7-methylguanosine linked via a unique 5' triphosphate bridge to the first nucleotide of the mRNA. Despite its relatively small size compared to the entire mRNA molecule, it plays a crucial role in multiple aspects of mRNA metabolism and turnover. Aside from its fundamental role in protecting the 5' end of mRNA from exonuclease-mediated premature degradation, cap structure is also involved in regulating nuclear export, enabling efficient translation initiation, supporting splicing, and immune recognition. So, although small, the cap is absolutely essential for both the stability and functionality of mRNA. It is also a critical feature of all mRNA-based therapeutics. Furthermore, subtle modifications within the cap structure can be introduced to modulate the properties of the entire molecule, enhancing its stability and improving protein expression efficiency. But before all that, the cap structure must be incorporated into the RNA strand. In addition to the enzymatic method mentioned earlier, co-transcriptional capping has become the gold standard in mRNA manufacturing. Unlike the enzymatic approach, which requires a separate post-IVT capping step, co-transcriptional capping is a one-pot reaction that occurs during mRNA synthesis. The cap analog is added to the reaction mixture together with the DNA template, NTPs, and RNA polymerase, resulting in capped mRNA product. Looking into the past, early co-transcriptional capping strategies relied on dinucleotide cap analogs. However, this approach had several limitations. The incorporation efficiency was quite low, leading to a high fraction of uncapped mRNA species. Additionally, cap analogs could be incorporated in the wrong reversed orientation, resulting in non-functional transcripts. This issue was successfully addressed with the second generation of capping reagents, known as ARCAs, anti-reverse cap analogs. The introduction of a small methyl group at the 2' or 3' position of the ribose within M7G prevents incorporation in the incorrect orientation. However, low capping efficiency remained a challenge, primarily due to competition with GTP for incorporation at the first nucleotide position. This limitation was overcome with the development of trinucleotide cap analogs, which provide significantly higher incorporation efficiency, often exceeding 95%, along with high IVT yields. They also better mimic natural eukaryotic mRNA and enable the introduction of Cap0, Cap1, or Cap2 structures. Currently, several cap analogs are available to choose from, and I would like to highlight one developed by ExploRNA Therapeutics. ExploCap is the co-transcriptional capping reagent designed as a cost-effective alternative to the current industry standard. It ensures high IVT yields and high efficient capping incorporation while offering broad compatibility across diverse IVT conditions and workflows, making it a versatile and plug-and-play solution for mRNA manufacturing. Let's now look at some functional data. In the study published in Nucleic Acid Research, mRNAs capped with ExploCap were evaluated in vivo in mice. Two models were used: firefly luciferase as a reporter protein and human erythropoietin as a more therapeutically relevant protein. mRNAs were delivered intravenously using 3 different lipid formulations. As you can see from the data, ExploCap capped mRNAs, shown in green bars, produced protein levels comparable to those obtained with unmodified standard Cap1 mRNAs, regardless of the LNP formulation used. This demonstrates that the technology is not only efficient in vitro, but also functionally effective in vivo models. In addition, ExploCap performs very well with modified nucleotides. Here, I present in vivo data for mRNA containing either standard uridines or N1-methyl pseudouridines. The results demonstrate that ExploCap maintains high performance with both nucleotide types, supporting modern mRNA therapeutic designs where modified nucleotides are often essential. And finally, let me briefly introduce ExploRNA capping technology as a broader platform. Beyond ExploCap, we offer additional alternative solutions tailored to specific research or therapeutic goals. Our tetranucleotide AvantCap Q family, with unique benzyl modification at N6 position of adenosine, provides higher translation efficiency without compromising IVT yield, capping efficiency, or ease of use. In addition, AvantCap Q2 is the first analog on the market to enable straightforward implementation of the Cap2 structure. This feature could be particularly valuable for scientific research, opening new possibilities for mRNA studies. Taken everything together, I hope that these results highlight the potential of our platform for therapeutic mRNA development, including vaccines and protein therapies. If you want to learn more about our technology, please visit our website.
Thank you, Marcelina, for this presentation. To come back to the mRNA produced at OZ Biosciences, the ExploCap technology provides us with numerous advantages. The capping efficiency with ExploCap is either better or as good as cap references such as Cap1 and CleanCap. Coenzymatically capping allows to remove one step during IVT and thus directs a quicker synthesis, meaning less degradation and higher production yield. This allows to obtain a better quality of mRNA and saRNA for an improved gene expression that makes ExploCap mRNA the perfect alternative to CleanCap mRNA. Finally, the take-home message is that before leaving our facility, our mRNA passes drastic quality controls. We assess mRNA integrity using two complementary analytical methods: fragment analyzer capillary electrophoresis, which provides a quantitative assessment of mRNA size distribution and detects any degradation or truncation, and HPLC, which separates intact mRNA from impurities at high resolution. When possible, especially for catalog mRNA, the functionality is verified through cell transfection assays. As mentioned previously, our superior grade quality control includes the measurement of dsRNA by ELISA, since it is a critical immunogenicity parameter. Our products consistently fall below the detection threshold of 0.05%. Finally, we perform full sterility testing for all of our mRNA with additional endotoxin measurement for superior grade mRNA, ensuring that every mRNA product is pure, functional, and safe.
Okay, so thank you everyone for your attention. If you have any question following this webinar, you can contact us at contact@aussiebiosciences.com. And if you have any technical question, you can contact Cedric at tech@aussiebiosciences.com. As I said previously, please stay until I end the webinar so you can fill out the survey. Thank you very much. Have a nice day.
Thank you as well.